Okay, so hello everyone. My name is Yanis, and today I'm going to talk to you about GORi. Right after crocodile, it's kind of funny, but yeah, GORi is a work we have conducted with Patricia Thébault at the Computer Science Laboratory of Bordeaux and Fleur Mougin at the Bordeaux Population Health Laboratory. And the main idea behind this tool is to characterize a gene by using multiple controlled vocabularies at once. A gene set, sorry. So, as most of you know, we can characterize the biology behind the gene set by using multiple hierarchical and curated vocabularies. Perfect. Thank you. So yeah, basically there are a variety of vocabularies. The most well-known one is gene ontology, to characterize, for instance, the biological processes of the gene products. But if we're interested in diseases or cell types, we can use a different combination of vocabularies and gene annotations. And so the main question we are asking ourselves is how to characterize a gene set by integrating these different vocabularies at once. And to do that, we feel like our intuition is to identify relevant associations between vocabularies on the basis of a specific gene set. So here I'll define a relevant association as a strong and informative association. And our hypothesis is that if two terms from different vocabularies are strongly associated together, then we expect them to annotate the same genes. There is an obvious limitation because the gene annotations are heterogeneous. But if we accept this hypothesis and this limitation, we can follow the pipeline I'm going to show you. So the first question, if we accept this, is to quantify the strengths of associations. And to do that, we use a framework called frequent item set mining.
I have a problem with that.
Oh, okay. It's perfect because I will take a lot of time on this slide, so you have the time to integrate it.
Sorry. Yeah.
Okay, so I'll try to look at it like that. Yeah, to resume, as I was saying, we follow a framework called frequent item set mining or association rule mining. You might have heard about it before. The main intuition behind it is to look at a database of transaction and to identify when items co-occur. For instance, that is used in supermarkets. If you consider that a client is a transaction and that the articles inside of their basket are items, you can use this algorithm to identify items that are most often bought together. A common example is chips and beers and beer. Yeah. And our intuition was to follow this framework, but adapt it to our gene set. Basically, here we will consider every gene in our gene set as a transaction and every annotating term of this gene as an item. And we will quantify the strengths of the association by using a metric developed in this framework. It's called the lift. The idea is that you look at the frequency of occurrence of A and B in your transactional database compared to the expected frequency if they were independent. Basically, if you have a lift of 4, in this example, it means that A and B are paired together 4 times more often than you would expect if they were independent. Okay. Oh, so I need to do that. Okay, perfect. So with this framework in mind, the first step of this work was to create a transactional database based on these hierarchical vocabularies and these gene annotations. And to do that, basically, we took different biological thematic characterization existing. So the gene ontology for biological processes, we took the mesh for diseases, and we've integrated these different resources into a single data warehouse. I might go back to it, but basically what's really nice here is that you have the versioning of each knowledge base that you're using, because the data warehouse is updated if you want to. And to illustrate how the pipeline works, I'm going to show you how it works when we take 81 genes that were differentially expressed following a vaccinal treatment. And here we will try to characterize this gene set based on the cell types and the biological processes that you can identify in this gene set. So the first step here is to look at the cluster map on the left. On this cluster map, every row in blue is a biological process, and every column in orange is a cell type, and the color of the cluster map corresponds to the lift value. So the lighter the color map is, the higher the association between this biological process term and the cell type term, the higher the association is. And here you can see real quick that there is variability in these associations, but there are some patterns that can be seen. And after the step, we end up with more than 10,000 associations. So now our next question is, how can we identify within these the strong associations? And to do that, I won't really go into the details, but we use independency tests, and we use them as a heuristic. If you want to know more about the heuristic, come to the poster. But basically, the idea of an independency test is to input in a contingency table the number of genes annotated by A and B, the number of genes annotated by neither, and the number of genes annotated by only one of them. And here we want the green diagonal to be much, much stronger than the red diagonal. And if that's the case, we can reject the independency of A and B. Okay, so with that in mind, we end up with more than 100 associations, and we want to further filter them to identify the most informative ones. Here, the idea is really naive. We simply use the hierarchical relationships of the different ontology we use to prune the redundant associations. So basically, if we have an association between T cell and immune response, and another one between biological process and the cell type, obviously we will keep the former one because it's more informative. This is the result shown in a graph for this one toy example. Basically here, each node is a term. It's color-coded in orange if it's a cell type, in blue if it's a biological process. And what's really interesting in the first part is that all of these terms are relevant, more or less, to the immune response. And if you remember, our initial gene set was identified following a vaccinal treatment. And the most important part, because here we characterize the gene set based on the edges, it is that the edges are relevant biologically. So for instance, here you can see that B cells and Paneth cells are involved, the genes that are marker genes of Paneth cell and B cells are involved in peptide production, and biologically it's a known phenomena. These results are really interesting, but they can be more interesting if we take into account different vocabularies. For instance, I was pretty surprised, but when we look at symptoms with a human phenotype ontology, we can identify associations between pathways such as the MAP kinase and the vertigo, arthritis. And after, when you look at the literature, there are recent articles that are trying to investigate these relationships. So here, the main idea is that these results can be really usable, but there are a lot of perspectives. Mostly the one I'm the most interested in is to improve the visualization aspect by integrating the hierarchy inside of the results. Right here, it's pretty lackluster. And also, it will be really great to integrate the literature directly in the pipeline to facilitate this curation step. And yeah, this was about the work in itself, but the main messages I wanted to share during this presentation, there's two of them, and three actually. I'll go back to the third. Basically, association mining is a really nice tool to quantify the relationships between qualitative features. And I feel like it can be really relevant in multiple case studies. For instance, in my work, I'm using it to integrate clusters together, so nothing to do about gene annotations. And another point I wanted to address is the fact that in biology, it can be really relevant to use these kind of approaches because they really help bridge the gap between reductionistic information and holistic information. Here, when you look at a gene set, you can have information on the diseases level, but also the biological process level. Yeah, these are 2 points I wanted to address. A third one that I haven't introduced in the slides because I felt like I was a bit illegitimate, but I'll talk about it anyway, is the fact that by using this kind of approaches, I'm not trying to promote the tool, but these kind of approaches are very easy to reproduce because, as I was saying, the data warehouse is based on versioning, and every analysis is conducted within the data warehouse. And there was a really interesting article by Mark Zeman's lab. I don't think people have read about it. That was mainly focused on gene set enrichment analysis, and it has shown that when you look at it, more than 95% of gene set enrichment analysis can be reproduced, and we really need to address this issue, I feel like. And here, I'm not saying let's replace gene set enrichment with this, but I want to say that going forward, it would be really nice to keep that in mind. And yeah, basically, these are the main points I wanted to address. Thank you for your attention. I don't want to be impolite, so thank you as well for the invitation, and thank you to my collaborators who could not be here today. And I don't really remember the number of my poster, but if you want to talk about, everyone is in the same alley for the bioinformatics in France, so it should be okay. And yeah, if you want to discuss gene annotations, single-cell transcriptomics as well, and the overall scientific discussion, I'm more than welcome to talk about it with you. Thank you.